To understand the predicament faced by humanity in developing effective therapeutics against deadly diseases, one need not look further than the COVID-19 pandemic waging around us right now. You might have often heard that it takes at least 10 years for a drug to be discovered and commercialized. The bitter truth, the actual journey might even take 3 times this time period. So what could be done to accelerate this tasking process? Hailing from the foothills of the Eastern Ghats, washed by the green waters of the Bay of Bengal, the best kept secret of India, a modest city is a microcosm of the future that we hope to contribute towards to make a better world. We are iGEM IISER Berhampur, and this marks our maiden venture at iGEM, where we are aiming to do just that.
To address the pressing issue of drug development, we at iGEM IISER Berhampur decided to look more closely into the dengue disease. We chose this because we needed a concrete problem to work on, something that we have seen wreaking havoc all around us, and yet does not have any effective drug or solution till now. Dengue, an infectious disease characterized by high fevers, headaches, rashes, and pain, is usually not fatal but can be very acute and unpleasant. Also dubbed as breakbone fever, dengue is caused by the dengue virus, which belongs to the Flaviviridae family and is carried by female Aedes mosquitoes. In most cases, recovery occurs in a few weeks. However, for a few unfortunate souls, it can be life-threatening. Severe disease can happen with any dengue infection, but is generally more common during a second attack.
The disease is a critical socio-economic problem in the tropics and subtropics. The virus shows a diverse clinical picture, making accurate diagnosis difficult. The existence of 4 different dengue serotypes and a lack of effective commercialized antiviral drug or vaccine makes it a difficult disease to tackle. Supportive care with painkillers, hydration with fluid replacement, and sufficient bed rest are the only accepted management strategies, and the final nail in the coffin is revealed by the ominous statistics. The incidence of dengue has increased 30 times over the last 50 years. Up to 390 million infections are estimated to occur annually around the world, putting over half of the world population at risk. The surge in cases are attributed to several factors, including increased population growth rate, global warming, spike in urbanization, and lack of healthcare amenities. A lack of awareness on the part of the masses is reflected in poor and inefficient mosquito control programs, which aggravates the issue. To get a glimpse of what people know about dengue fever, and also whether they know people who have been infected by the disease, we conducted a survey in our country. We collected more than 700 responses, of which more than 92% were aware of the disease, out of which 65% admitted to taking precautionary measures against the spread of the infection. Even though the distribution of the responses we received was biased towards certain states, it dawns clear that dengue is a serious contrary in our country. To gain a quantitative understanding of the severity of the problem, we looked into the epidemiology of dengue. By mining data from several published papers and national archives, we were able to examine past trends and the increasing incidence of dengue fever. Comparison of the epidemiological data with other arboviral diseases ascertained that dengue, unlike others, is following an upward trend in India. After gaining a comprehensive understanding of the past, we forayed into the future. To see how serious the issue is, we tried predicting the burden of the disease by using a modified SIR plot. Our model accounted for relevant variables and was fitted with the current dengue-infected data of India obtained for 23 years. The complete plot for the whole population of India depicted that the disease harbors a potential to infect 4 lakh people per year by 2025. With this, we safely concluded that this is a problem that needs to be addressed.
Now let's look at our game plan. We have decided to come up with a 2-pronged approach to provide a solution to this problem. Firstly, in the essence of iGEM, we will be using synthetic biology to partially replicate the infection state where the virus interacts with the host system to gain entry and invade the tissues. So even without using cells isolated from patients, we can create an artificial system that will enable us to study the components of our choice reasonably. For this, we are creating a modular reporter system consisting of viral components and host components fused to fluorescent tags and assembled in a mammalian expression system. These modules, assembled using synthetic biology tools, will contain bacterial, human, jellyfish, and viral sequences. The interaction will be modulated by the use of chemical biology tools. This interdisciplinary approach will provide us with FRaPPE, a FRET-based rancor for proteins and peptides. Our second approach is to tackle the social aspect of the problem by creating awareness about the disease, the causes, and prevention strategies by reaching out to the community at large.
But coming back to FRaPPE, let us take you through our design process. We wanted to solve the dengue scourge. So what do we use as our weapon? What is so special about this virus that can give us an edge over conventional supportive therapies? As it turns out, dengue is one of the masters of an intricate process named protein-protein interactions or PPIs. In this phenomenon, two complementary proteins in close proximity come in contact with each other in a certain orientation, forming a complex which is either transient or permanent in order to perform a specific function. In a cell, interactions between various proteins can bring about numerous cellular processes and pathways. Especially during a viral infection, the virus can only survive and multiply in the host system by making use of numerous protein-protein interactions between its proteins and proteins of the host cell. Thus, inhibition of host virus PPIs is a therapeutic target that holds potential in order to develop an effective antiviral drug against dengue and all other viral diseases where this is a common theme. Our reporter system FRaPPE uses two techniques to study the protein-protein interaction of interest. One is chemically induced dimerization, wherein two particular proteins which can be induced to dimerize with one another are expressed in a complex with our selected proteins. In this way, our chosen proteins can be made to interact by bringing them in close proximity. In our system, we aim to use FKBP12 and FRB protein domains to act as the modules that regulate the interaction. These are induced by the drug rapamycin, the chemical inducer of dimerization. With the CID reporter, we can ensure that our chosen PPI takes place in the cell and we can localize and observe it more closely.
To quantify the extent of the interactions, we are using a FRED reporter. FRED is a phenomenon that essentially functions as a spectroscopic ruler by employing two fluorescence proteins wherein the excitation energy of the donor is transmitted to the acceptor in its vicinity. This results in the distinctive fluorescence emission spectra of the acceptor. The appeal of using these biosensor systems is their high sensitivity to the distance of separation between the interacting molecules. Due to this property, FRED is used to quantify many biological processes that are dependent on molecular proximity, such as protein-protein interactions. It is more advantageous than other reporter systems because it allows the observation of live cells in a non-destructive and minimally invasive way. To implement the system, the FRaPPE constructs as plasmids can be cloned into the chassis E. coli to first select for positive recombinants and then transfected into the second chassis, human embryonic kidney cells, to allow observation in the mammalian cell line. The combined use of CID and FRED will thus give a robust, high-throughput screening system for inhibitory drugs. By comparing the FRED readout obtained as the efficiency between two conditions, namely without rapamycin and with rapamycin, we can analyze the strength of the interaction. Upon administration of the inhibitor drug, the PPI will be attenuated, and if the inhibitor works, this can be quantified by the change in the FRED efficiency. Ideally, based on the ability of the inhibitor to break the protein-protein interaction interface, the FRED readout should resemble the scenario when there is no rapamycin in the system, effectively serving as an internal control.
So we now know how FRaPPE works. The real question was whether this would indeed be a useful addition to the researcher's toolkit working on combating infectious diseases. This is where our problem of choice, dengue, came in to help give the proof of concept. We first needed to address the question, which of the myriad PPIs used by dengue should we focus on? Antiviral approaches explored thus far have targeted both structural and non-structural proteins of DNV, and we decided to go for the latter category. To justify our choice, we looked into the mutational landscape of the 4 serotypes of the dengue virus. We compared the number of mutations that each of the genes have gone through and found that the structural proteins, despite having less number of amino acids or a shorter chain length, have a higher probability of undergoing mutations, thus rendering the inhibitor ineffective. In fact, the C and E proteins undergo a significant number of mutations in all the 4 serotypes. So for designing the inhibitor, NS proteins remain the more sensible choice as they might be exempt from background evolutionary selection pressures. Now the dengue virus boasts a repertoire of 7 non-structural proteins. They are the workhorses for viral replication and can also temper host immune responses in clever ways. An extensive literature survey revealed the complex DNV-NS interactome. By analyzing the amount of structural and experimental data that was available for each PPI, we narrowed down our target to the NS5 protein and one of its host partners, STAT2 or signal transducer and activator of transcription.
We will touch upon a few characteristics about our PPI of choice and why it can be a suitable target interaction for designing a novel therapeutic. When the dengue virus infects a host cell, various pattern recognition receptors within the cell bind to the RNA molecules, triggering parallel signaling cascades. A leading player here are proteins known as interferons, which establish a non-permissive state in uninfected host cells. The virus has thus evolved mechanisms to antagonize these defense pathways. Type 1 IFN signaling, when blocked by the viral maneuvers, can attenuate the antiviral immune response. This cascade is mediated by the JAK-STAT pathway, and NS5 targets STAT2 for ubiquitin proteasome-mediated degradation, thereby blocking it. If your cells do not have functioning STAT2, the IFN response is compromised, promoting viral spread.
Once all these initial questions are answered, we entered the Drylab. Our target was to design peptide inhibitors interrupting the NS5 STAT2 interaction using various bioinformatics and in silico analysis. First, using the structures of NS5 and STAT2 and experimental data available about their interaction, we designed a model for NS5 STAT2 complex by molecular docking. Then, using this structure as a template, we generated potential peptide sequences which are able to bind to those residues on NS5 which are involved in its interaction with STAT2. Afterwards, based on data available from literature, we modified the sequence a bit to account for the stability and solubility. Once this was completed, we screened the resulting peptide library by docking them against NS5 and eventually narrowed down to a few potential peptide inhibitors for in vitro screening using FRaPPE. This process of generating peptide inhibitors was carried out with the guidance and help from Dr. Malay Kumar Rana, an expert in computational chemistry from iGEM IISER Berhampur.
To design our wet lab constructs, the docking experiments were the starting point. Using the visualization of the structure based on the experimental model, we chalked out a rough design for our plasmids. But since the experimental data was limited and did not resemble the entire length of the proteins, we performed homology modeling with the primary sequences of the proteins of interest to obtain a clearer picture. This reconstructed model provided us with insights about selecting appropriate N or C terminals to position our FRET fluorophores. Interviews with experts in this field, Dr. Vinay Bulusu at IISER Berhampur and Dr. Pooja Singh at Center for Cellular and Molecular Biology, Hyderabad, helped in further refinements to the design. We were able to understand how the FRET readout could function as the screening tool, how to ensure that the translated proteins in the cells would satisfy optimum separation distance to ensure FRET, importance of the controls, and even ideas to explore alternate ways to introduce our plasmids into the chassis and ensure stability. The constructs we planned contained coding sequences of our proteins tagged with fluorophores and modulatory CID domain sequences in different combinations. Control constructs with fluorophore tagged CID domains alone and with fluorophore tagged with the coding sequences of the proteins of interest alone were also included. All of these, once generated, will be contributed to the iGEM repository as our FRaPPE system.
While we were waiting for our CID FRET plasmids and necessary reagents to get delivered, we have been running optimization experiments. The plasmid being used for this contains sequences for fluorescent proteins M turquoise and CP173 Venus and is being confirmed running different sets of restriction digests to be followed by transformation into competent DH5 alpha E. coli cells and transfection into each HEK293 mammalian cells. Fluorescence observations for the positively transfected cells in the ongoing experiments will enable us to optimize the protocols for generating our mammalian reporter.
Simultaneously, while working with the in silico peptide inhibitor designing pipeline, we worked on molecular dynamic simulation for the NS5 and STAT2 interaction to gain more insights on how these two proteins interact. The planning and execution of the MDS experiments were performed with the continuous support from Dr. Sunita Patel from Center for Basic Sciences, who is an expert in molecular modeling.
We have been talking a lot about performing modeling studies to determine the interaction between our proteins of interest. But how do we use FRaPPE to gain information about the peptide inhibitor efficiency? The process involves titrating our cell culture with the peptide inhibitor and measuring the FRET emission. We will get an emission versus log of the concentration curve, from which we will obtain IC50 values, which is just a fancy name to describe the concentration of the inhibitor at which the emission becomes 50% of the maximum value.
As of now, we have designed our FRaPPE constructs and are optimizing our transfection protocols. Once we have our FRaPPE T2 and T3 co-transfected cells, we will do a dose assay for finalizing an optimum rapamycin concentration. Excitation of CFP would yield a certain FRED efficiency. In the context of infected cells, this resembles a block in the downstream interferon pathway. Upon adding one of our designed IPEPs, the FRED efficiency would be different. The downstream interferon pathway would thus be free from inhibition. Our IPEP library, doc models and MDS results will be contributed to the iGEM registry. We expect that once our biobricks are ready, they will also be contributed as parts to the registry. In a nutshell, our project will eventually result in the construction of a mammalian reporter system that can mimic viral protein interaction with hosts or the viral proteins in an exogenous system. Although we proposed this for studying dengue interacting proteomes, the reporter can be easily adapted to any viral protein including SARS-CoV-2. Additionally, the project will also result in validation of peptide inhibitors as a potential intervention strategy for dengue infections. This methodology will thus pave new vistas for antiviral research, acting as a rapid drug screening module for therapeutic interventions.
Dengue is a serious problem, but people might not be taking it seriously. We met with a doctor who works in a dengue-prone rural area and found out that awareness is a key aspect in mosquito-borne diseases. So we decided to conduct awareness programs at two levels. One, through our social media, where we shared posters and threads regarding the prevention of mosquito-borne diseases and also the various symptoms of dengue. On the other level, we organized a crowdfunding campaign to help us provide mosquito nets, masks, and information brochures to people living in low-resource areas and lacking hygienic healthcare amenities. Fortunately, we were able to collect around 60k INR, which is around 800 USD, for this cause, through which we distributed the amenities to 70 families in Berhampur. We also got in touch with local workers who craft mosquito nets for a living and even got a chance to help them during this unprecedented pandemic by purchasing these nets on a large scale for our cause.
We believe that science not communicated is science conspired. In collaboration with Team iGEM IISER Pune, we conducted an interactive online workshop for middle school students on the prevention of mosquito-borne diseases. Our efforts did not only end there. We contributed several articles spanning domains of synthetic biology to different established blogs and also designed pamphlets, posters, and comic strips that were published on our social media handles to reach out to people in all mediums possible. An online forum Scintillate was also created by us, where we engaged enthusiastic young writers from different disciplines in our home institute by compiling their articles. This was done with the aim of generating a resource that can help individuals from different walks of life to get a flavor of this diversely exciting field. To give a glimpse of infectious diseases and how pathogens can be both good and bad, we created a coloring book for children, Splash. This offered the kids a fun and engaging way to learn about science. It was accompanied by a Q&A forum where we addressed the questions of all the inquisitive young minds, trying to do our part in kindling their curiosity.
The restrictions of online mode encouraged us to find unique ways to collaborate and connect to the iGEM community this year. From hosting the All India iGEM meet with fellow iGEMers, then the morale boost we had from securing second position in project presentation pitch at the worldwide virtual Parisian meetup to having meaningful collaborations with a number of teams, there was nothing more we could ask for. Starting from iJet to iMask, we certainly had a blast. However, our journey might not have been possible without the help from our PIs, Dr. R. Selvi Bharadavikru and Dr. Sandeep Chatterjee. We will remain grateful to the staff, professors, and students of our home institute for their unwavering support in all our activities. We thank our sponsors, GenScript Molecular Cloud, Eppendorf, SnapGene, MathWorks, New England Biolabs, Twist Bioscience, and Integrated DNA Technologies, whose unflinching support even during these difficult times helped us reach where we are today. We feel truly fortunate to be part of iGEM 2020, and we hope to continue the spirit of research, teamwork, and collaborations in the coming years as well.